Hi, I'm Zari from Ace Academy, and today I'm going to solve 9700 October-November 2022 variant 11. So, without further ado, let's begin. Question number 1. The photomicrograph shows part of a plant cell. What is the magnification of the cell? So now, in order to tackle this question, we need to find out the formula, which is magnification equals to image length by actual length. Image length by actual length. So there is a bar given which shows us that this is equal to 5 micrometers, but unfortunately I can't really measure this since I'm solving this digitally. So I'm just gonna use a rule to measure this. It comes out to approximately 2 centimeters. So the actual image length is 2 centimeters by 5 micrometers. So we have to convert them to the same units because magnification is a ratio and ratios do not have any units. Thus, 2 into 10 to the power minus 2 meters by 5 into 10 to the power minus 6 meters. Since I like keeping my units in meters, it cancels out easily. So meters cancel out. And the answer is 4,000, which is option D. Now, question number 2. Which row matches each organelle with its function? So I really like this question because you can know a lot of things from this chapter, from these MCQs, because they have a lot of descriptive content, these tabular questions. So let's start. Option A, microtubules make cilia and spindle. Well, this is correct. This is a function. Rough endoplasmic reticulum separates some processes from cytoplasm. Well, this is true because inside the rough endoplasmic reticulum, modification of proteins occurs. So, modification of protein occur here, which is inside the rough endoplasmic reticulum, and it separates these processes from the cytoplasm. So this is correct. So Golgi body, yes, this makes lysosomes, correct, because the vesicles which are formed from the ends of the Golgi body can be used to make lysosomes. And then centrioles, yes, assemble the spindle during nuclear formation, yes. As we read in chapter 4, the centrioles move towards the opposite poles and then they make spindle fibers which then pull the chromosomes towards the metabolism plate and then eventually towards the poles during anaphase. So this is correct. And then, so I think option A is already correct, but we are going to look at the other options as well. So option B, move vesicles within the cell. This is correct because one of the functions of microtubules is it provides mechanical support. Mechanical support to the cell and forms the cytoskeleton. I'm writing this because this is good for revision and you can also know more about these functions which are not in this MCQ. So, forms part of the cytoskeleton and it forms flagella as well as centrioles. Okay, great. So apparently this is correct. And then rough endoplasmic reticulum, yes, assembles amino acids to make proteins. Yes, this is correct, as well as modification of proteins, as we saw in option A. Golgi body contains enzymes for destroying worn out parts of the cell. No, it produces. If it was written produces, then it would probably have been correct. And then centrioles move chromosomes apart during anaphase. Yes, this is correct, as I already talked about this. Now, since this has one incorrect answer, option B is incorrect. Option C, Move chromatids apart during anaphase. This is correct. Move modifies proteins which may be released from the cell. Yes, this is correct. Modification and synthesis of polypeptides. Golgi body makes glycoproteins. This is also indeed correct. Yes. Then centrioles. These move cilia. No, these form part of the cilia. Form part of basal body of cilia. The wording is incorrect and hence this option is incorrect as well. Now moving on to the last one. Microtubules form part of the cytoskeleton. Yes, here, as we talked about here. So this is correct. Makes triglycerides and phospholipids. Well, this is wrong because this is a function of the smooth endoplasmic reticulum, not the rough endoplasmic reticulum. Golgi body modifies proteins by adding carbohydrates. This is called glycosylation, and this is correct. The other functions of Golgi body include formation of vesicles from edges, then packaging proteins, then secretion of vesicles, and etc. etc. And finally, centrioles move individual cells. This is a big no-no because these move organelles within the cell through its cytoskeleton or move chromosomes during mitosis. So A is the correct answer. Now, let's move on to question number 3. Where are ATS ribosomes found? Option A, chloroplast, no. 70S. 70S ribosomes are found. Cytoplasm of eukaryotes. Well, this is correct because ATS are found over here. Mitochondria, 70S. Well, this is correct. And then cytoplasm of prokaryotes. This is 70S. So, the only option which is correct over here, which is number 2 and includes number 2, is option C. So C is the correct answer. Now, question number 4. Which feature, when present in an organism, enables it to be identified as a prokaryote? So, one of the key features of prokaryotes is cytoplasmic DNA, which is naked DNA and not associated with histones. So I'm guessing A is already the correct answer. However, let's cross out the other answers as well. Cell wall, no, this is incorrect because animal cells have, plant cells have cell walls as well. Nucleus, no, because prokaryotes do not have nucleus. And then ribosomes, well, archaea, bacteria, as well as animalia and plants have ribosomes. It's only the 70S and 80S that differentiates what ribosome it is and which to which family it belongs to. So D is incorrect. Number 5. So for 4, A is the correct answer. So number 5. What is the function of the nucleolus? The formation and breakdown of the nuclear envelope. Okay, yeah. What is the function of nucleolus is synthesizes tRNA, which is used to bring amino acids to a growing polypeptide chain in a ribosome. And another function is rRNA. Synthesizes tRNA and rRNA. rRNA is a substance which is used to make ribosomes along with ribosomal proteins to make the ribosome complex. So, option A, the formation and breakdown of the nuclear envelope, no, this is incorrect. The formation of rough RER, incorrect. The synthesis of ribosomal proteins, well, no. The synthesis of rRNA, yes, because this is a function of the nucleolus. So D is the correct answer. Number 6 is an analytical question, which is a food test. So, the diagram shows the results of a number of tests on a solution of biochemicals. So, which substances are present in the solution? So, Benedict solution is blue. So, its original color was blue as well. What does this mean? This implies that reducing sugar absent because blue to blue, the color remains unchanged. Non-reducing sugar test yellow, so the color changes blue to yellow. So there must be some non-reducing sugar present. So non-reducing sugar present. Number 3, iodine test blue-black. So yes, color changes well this time. Yellow to blue-black. So starch is present because this is the test for starch. And finally, biored test blue. So the color again remains unchanged. If protein was present, then it would have been purple, but it isn't purple, it's blue. So protein absent. So in this question, only starch and non-reducing sugar present, which gives us option A. So A is the correct answer. Now let's look at beta glucose. So in beta glucose, OH is always above the plane of the 6 carbon atoms, above plane of C atoms in carbon 1. So conventionally, carbon 1 is this one, then 2, 3, 4, 5, 6. So let's look at option A. Yes, this looks correct because it is above the plane. Then carbon 2 is always below, correct? Carbon 3 is supposed to be above, correct? 4, correct? So this is the correct answer. Then let's look at the other answers, why they are incorrect. So carbon 1 automatically already wrong, and the rest of the molecule looks right, but this is actually alpha glucose, so it's wrong. So C, this is above the plane, which is incorrect. It was supposed to be downwards. Then 3 is correct, 4 is downwards, correct. So this is correct, only that this is, well, a different form of glucose, so incorrect. Then finally, let's look at D. This is incorrect, obviously, because this is below the plane. And then this is below the plane, which is correct. This should be above, correct. And yeah, well, the only thing wrong with this diagram is carbon number 1 and carbon number 3. Otherwise, the molecule looks okay. But so, for question number 7, A is the correct answer. Now 8. Which diagram correctly matches the example with the type of molecule? So fructose is not a disaccharide, it's a monosaccharide, along with galactose and glucose. Okay, macromolecule glycogen, correct. Monomer glucose, polymer starch, yes. Just because the first one is wrong is the reason why the entire answer is wrong. Okay. Disaccharide. Starch is not disaccharide, is a polymer or polysaccharide. Macromolecule hemoglobin. Yes, this is correct. Monomer, ribosome, correct. Glycogen is a polymer or a polysaccharide. Yes, correct. So, maltose. This is indeed a disaccharide. Ribonucleic acid or RNA. This is a macromolecule. Monomer, yes, no, this monomer is not sucrose. Sucrose is a disaccharide, and polymer. Cellulose is a polymer, so yes. So again, this is the incorrect answer. So this automatically leaves us D for the correct answer. But let's look at it. Sucrose is a disaccharide. Cellulose is a macromolecule or a polymer. Fructose is a monomer, correct? And then collagen is a polymer, yes, because it is it's a fibrous protein. Okay, so that gives us D as the correct answer. Now let's look at question number 9. The enzyme alpha amylase hydrolyzes amylopectin, but it is not able to hydrolyze some of its glycosidic bonds. It only hydrolyzes one full glycosidic bonds. All right. So 1,6 it can't. 1,6 cannot. 1,6 glycosidic bonds. It is not able to hydrolyze the last bond of a chain. Okay, nice. It is not able to hydrolyze the bonds in a chain of 3 units attached by a 1,6 glycosidic bond to another carbon atom. So let's label these as 1, 2, and 3. Okay, so D is already incorrect because it does not satisfy 2. It is not able to hydrolyze the last bond of a chain, so this is incorrect. Then let's look at A. A is incorrect because it is not able to hydrolyze the bonds in a chain of 3 units attached by a 1,6 glycosidic bond. And B is incorrect because it is a 1,6 glycosidic bond itself. So that leaves us C to be the correct answer. So moving on, molecule X is a lipid. Okay, lipid, nice. Which row is correct for molecule X and a triglyceride? So let's look at here. This over here is the ester linkage. Nice. And this over here would be the carboxylic acid or the fatty acid, as we would say. So as we can see here, molecule X has one alcohol, one fatty acid. So this means, let's move on. Which row is correct for molecule X and a triglyceride? Molecule X contains one unsaturated fatty acid. No, all of them are saturated. Triglyceride contains such. No. Molecule X contains no ester bonds. No, because as we can see here, there is an ester bond. So this is automatically wrong. Triglyceride contains 3 ester bonds, correct. Molecule X contains one fatty acid. This is correct. As we see here, there is indeed one fatty acid. Then triglyceride contains 3 fatty acids. Well, this is correct as well. Molecule X contains 2 fatty acids. No, there is just one. And triglyceride contains 3 fatty acids. This is correct. Triglyceride has 3 fatty acids joined to a glycerol molecule. Something like this. So this leaves us as C as the correct answer. Okay. Moving on to question number 11. Bread contains a mixture of polypeptides known as gluten. 2 of the polypeptides found in gluten are glutenin and gliadin. Which statement describes the tertiary structure for protein? So, tertiary structure refers to 4 things. Firstly, ionic bonds, which happen between charged R groups. Then hydrogen bonds, which happen between polar R groups. Then disulfide bonds, which form between only cysteine amino acids. And finally, there are hydrophobic interactions, which occur between non-polar side chains. All of them follow side chains or R groups. Hydrophobic interactions. So let's look at the options now. While tertiary secondary structure occurs between the repeating CO and NH sequences that form the amide linkages of the peptide bonds of the polypeptide. So del plus, del minus, del minus, del plus, then an adjacent hydrogen atom will, from another amino acid which is parallel to it, will form hydrogen bonds because the positive and the negative will attract each other. This is how hydrogen bonds in secondary and tertiary would be formed. Okay, moving on to the options. Disulfide bonds form between glutenin and gliadin. No. Disulfide bonds do not occur between polypeptides. It occurs between only cysteine amino acids, so it's wrong. A large proportion of amino acids in gliadin are glutamate. So what? It isn't talking about any property. No property. So it's incorrect. Alpha helical sections are found in glutenin and gliadin. Well, this is secondary structure, not relevant over here. And finally, Amino acids with hydrophobic R groups are found on the inside of the glutamate. Well, this is correct because this talks about tertiary structure. So D is the correct answer because the hydrophobic R groups are formed inside the polypeptide while hydrophilic are outside the polypeptide. So and D is the correct answer. Now, moving on to question number 12. A student was provided with two test tubes, one containing 10 cm cube of solution P and one containing 10 cm cube of solution Q. When these two solutions were mixed together and left for 24 hours, the concentration of P decreased, but the concentration of Q remained the same. The student wrote this formula in cases. P may break down over time. Well, this is correct. This may be correct because the concentration decreased. Q may be a biological catalyst, which is why its quantity remained the same. Okay, while P may be the substrate, this may be correct because P may be catalyzing P, which is why the concentration of P is decreasing over time. So also may be correct, which is why 12A is the correct answer. Moving on. Which statements are correct for the locking T hypothesis and the induced fit hypothesis of enzyme action? So in locking T, the lock is the enzyme and T is the substrate. While in induced fit, this is basically the same, but only that enzyme or substrate may change shape because it's not exactly complementary to the active site. So the substrate, option 1, the substrate is the same shape as the active site. Well, this is not correct because shape for induced fit might change. It's not exactly complementary. Okay. Now, this sentence is wrong. Number 2, the substrate is held in cases in the active site by temporary bonds. This is correct. The enzyme and sometimes the substrate change shape slightly as the substrate molecule enters the enzyme. This holds true only for induced fit, not for lock and key hypothesis. So 1 and 3 are incorrect, which leaves us with 2, and thus C is the correct option. Question number 14. A student investigated the rate of enzyme activity with increasing substrate concentration. The experiment was repeated with the addition of a reversible non-competitive enzyme inhibitor. A graph was plotted to show the result. Which graph represents the results of the two experiments? Well, in non-competitive inhibitor, it binds to enzyme, so Vmax is not achieved. So if the Vmax was supposed to be this, the Vmax with non-competitive inhibitor would be this. Vmax would not be achieved. So the dash line will be correct only, which is why B is the correct answer. A doesn't even make sense because why would it increase exponentially after a non-competitive inhibitor is added? It is incorrect. C, the rate of enzyme Vmax won't be higher with non-competitive inhibitor. So this is wrong as well. And then this is correct, but this is a competitive inhibitor. The competitive inhibitors reach Vmax, but after a certain concentration. So this is competitive inhibitor. And thus, B is the only correct answer. Now, number 15. A cuboidal epithelial cell has a length of 2 cubic meters, micrometers. The cell is an approximate cube shape. With what is the most accurate estimate of the surface area to volume ratio of this cuboidal epithelial cell? Well, a cube has 6 sides. So surface area equals to 6 into length into breadth. 6 into 2, which gives us 24 square centimeters. Then volume is 2Q, which is 8 cubic centimeters. Then SA is to V ratio would be 24 is to 8, which is 3 is to 1. So D is the correct answer. Now, let's move on to number 16. Which statement is correct for facilitated diffusion and active transport? So what is facilitated diffusion? The movement of molecules down a concentrated gradient through carrier proteins or channel proteins. And active transport is the movement of molecules against the concentration gradient using energy, and they also require carrier proteins or channel proteins. So look at option A. As the direction of the concentration gradient changes, so does the direction of the movement of the molecules. No. The facilitated diffusion and active transport have a definite concentration gradient. Facilitated diffusion is always down a concentration gradient, while active transport is always against the concentration gradient. So it's incorrect. Molecules always move at the same time as simple diffusion. Not really. For facilitated diffusion, and no for active transport, because active transport depends on the number of carrier proteins as well as the number of ATP molecules hydrolyzed to provide the energy for the movement of molecules against the concentration gradient. So this is incorrect as well. Number C. Specific molecules are transported across a membrane. This is true because molecules which are complementary to the shape of channel or carrier proteins are transported by facilitated and active transport. So C is correct. Number D, the molecule ATP is required to move specific molecules quickly through proteins in the membrane. Well, facilitated diffusion does not require ATP. It's a passive process, so it's incorrect. So C is the only correct option. Number 17. Equal-sized potato pieces were placed in a tube and covered with a sucrose solution. The test tube was left for 30 minutes. All other variables were standardized. After 30 minutes, the potato piece had not changed in size. Okay, this means water potential is same. It's in equilibrium, which means there is no net movement of water, but yeah, there is movement of water, just no net movement of water. Equilibrium, and you have to understand that the concentration of the sucrose solution after the potato solution may not be the same. And this is a common mistake. Common mistake that you need to know. Okay, what can we conclude from this result? The concentration of sucrose is the same in the potato and in the solution. As I already told, the concentrations are not the same, and there is no more movement. No, water does move. It's just that there is no net movement because it's cancelled out. So it's totally wrong. The concentration of sucrose is same in the potato. Again, the concentration may not be the same, and there is no net movement of water in the potato. Well, the second statement is correct, but of course it's wrong. But overall, these incorrect. The water potential is the same in the potato and in the sucrose solution. Yes, this is correct. And there is no more movement. No net movement. So this is incorrect as well. Then D. The water potential is the same in the potato and in the sucrose solution, and there is no net movement of water into or out of the potato. So D is the correct answer. Number 80. How many of these processes produce genetically identical cells? Growth of plant roots, yes, it produces a lot of cells which are genetically identical. Replacement of dead human skin cells, correct. Asexual reproduction of coral, yes. Repair of damaged muscle tissue, yes. Since it's talking about tissue, tissue needs a new cell. So this is correct. If it was talking about cells, then it would not have been correct. Since it's talking about tissues, is correct. So for all four of these processes produce genetically identical cells. So D is the correct option. Question number 19. Which row shows the correct number of each component of a single chromosome at the end of prophase of mitosis? So, firstly, let's draw a diagram of a chromosome at the end of prophase. So, these two are sister chromatids. So, as you can see over here, or two sister chromatids, there are four telomeres. There is just one centromere, and one chromatid has one DNA strand. So in total, there are four polynucleotide strands. So let's look at the options over here. Centromere, 1. Chromatids, 2. Polynucleotide strands, 4. Telomeres, 4. So for 19, B is the correct option. Moving on, question 20. Which processes occur during the interphase? DNA replication. So in interphase, there are three phases. G1, gap1, S, which causes DNA replication, and then G2. So DNA replication is correct. Mitotic organization, this occurs only in mitosis, and mitosis does not occur in interphase, so it's incorrect. And then synthesis of ribosomes, yes. Growth, then production of organelles, protein synthesis, and stuff like that, all of this occur throughout interphase. So 1 and 3 are the correct options, which gives us C. Moving on, question number 21. Embryonic stem cells are able to replicate continuously. So since they are able to replicate continuously, they are like stem cells. And normally, as you already read in chapter 5, telomere length decreases with each replication, and eventually the telomeres would grow so short that the cell will even die or replication might stop. So, in order for them to replicate continuously, telomeres must stay the same length, and this is done by the enzyme telomerase. Telomeres must stay the same length, and this is done by the action of telomerase. So, B is the correct answer. Number 22. Which nitrogenous bases are pyrimidines? So there are two types of bases. One is purines, another is pyrimidines. So the bases adenine and guanine, these are purines, while cytosine, thymine, and uracil, these are pyrimidines. And these are two carbon rings, and these are one carbon rings. So purines are bigger than pyrimidines. So anyways, this one is talking about which are pyrimidines. So thymine and uracil are pyrimidines. So C is the correct option. Number 23. DNA replication in bacteria were investigated. Bacteria were grown in a medium with only heavy nitrogen, 15N, until all the bacteria DNA was heavy. So this is a classic Michelson style experiment, and this comes a lot in MCQs. So this is how I like to approach these types of questions. So I I think of all the polynucleotide strands with 15N as blue. So since they were grown in 15N, all the DNA nucleotides have 15N. And then these bacteria were moved from this heavy nitrogen medium and cultured in a medium with only light nitrogen, 14N. This formed the first generation. So F1, in F1 to for DNA synthesis to occur, they must be separated and semi-conservative DNA replication occurs. So since there are no 15N, DNA has to be produced with 14N. So it is produced, thus producing hybrid DNA. And then the bacteria will continue to reproduce, and a sample of bacteria were collected from the second generation, and the DNA was analyzed. So again, they reproduce. the 14N polynucleotides are denoted by the red ball. So as you can see here, with each generation, the heavy nitrogen DNA continuously, the proportion of heavy nitrogen DNA decreases, while the proportion of light nitrogen DNA increases. So over here, 100% have at least one light DNA, 25% or 2 out of 8 or 25% have heavy DNA, are heavy strands actually, not heavy DNA, are heavy strands, and then 2 out of 4 or 50% are hybrid molecules. And hybrid refers to these molecules. So let's look at the options over here. Percentage of heavy DNA strands, it's 25%, as we already talked about, and percentage of hybrid DNA molecules is 50%. So option B is the correct answer. Now, number 24. The sequence shows a series of bases at the start of the DNA, TSC, and blah blah blah. After transcription, the mRNA was translated via tRNA into a sequence of amino acids. When part of the polypeptide was analyzed, was found to contain the amino acids in the table. So alanine 2, glycine 3, methionine 1, valine 1. Okay. So mRNA, this always has methionine in the front. So this TAC must be methionine. So this is MET. Oh, all the options have methionine. Okay. Then there are 3 glycine molecules. So CGA, oh no, CCA, CCA, CCA. So this is gly, this is gly, this is gly. Now, alanine has 2 molecules. So this must be alanine, this must be alanine, and what's left is CAA, which must be valine. So valine. So let's look at the options over here. Methionine, alanine, glycine, valine, glycine, alanine. So B is the correct option over here. Number 25. The table shows the mode of action of 2 antibacterial drugs that can excite the synthesis of protein. Mode of antibacterial drug, rifampicin. Mode of action, binds to RNA polymerase. So if it binds to RNA polymerase, no mRNA is produced. Okay. Then streptomycin. It causes errors in translation. So as a result, faulty protein will be produced because the wrong amino acid is added. Okay. If bacteria are treated with the drugs rifampicin and streptomycin, what will be the immediate effect? Transcription will stop, but faulty proteins may continue to be synthesized because of mRNA that was already produced before rifampicin was added. All right, so this is correct. If translation has started, proteins may be faulty. This is due to the action of streptomycin. This is correct. Translation will be inhibited. Is it? This is wrong because there is nothing about inhibition in the mode of action of streptomycin. So 1 and 2 is correct, which gives us B as the correct option. Okay. Number 26. This is an electron micrograph. Which structure contributes to mass flow by the use of proton pumps? So, in a leaf, xylem is above while phloem is below. So this big tube over here, this must be xylem. So this is incorrect because xylem does not use proton pumps. Then D is epidermal cells. Again, this is incorrect. Then C, this is phloem and this has companion cells, and companion cells have proton pumps. So C is the correct option. And B is a hollow space. This is not even a cell. So B is incorrect as well. Okay. So B is the correct. C is the correct option for 26. All right. So moving on to question number 27. The diagram shows some tissue types in plant organs. So number 1 is a dicot root, while number 2 is a dicot stem, and number 3 is a dicot leaf. So as we already talked about in the last MCQ, number 4 will be xylem, while the lower part will be phloem. As you can already saw what we inserted in question number 26. And then in dicot root, the star-shaped portion is xylem, while the peripheral features are phloem. And in number 2, the peripheral ones are phloem, while the part to the left or to the inside of the cambium is the xylem. All right. So 1 is phloem, 2 is phloem as well, 3 is phloem, while 4 is xylem. So overall, A is the correct answer. Okay, so let's move on to question number 28. Which description of xylem vessel elements is correct? Cells join to form a tube. This is correct. Pits at intervals, correct. C, plates between cells, no. It's free. Between the cells, there are no end walls. So this is incorrect. And it's surrounded by endodermis and root. Well, not really. Well, correct. Okay. But overall, it's correct. Xylem contains cells joined end to end. This is correct. Containing cytoplasm. Correct. No, it doesn't contain cytoplasm. And cell walls with lignin, correct. And located to the outside of phloem in vascular tissue. Yes, it is indeed correct. So, cell B is incorrect. Xylem contains elongated cells with end walls broken down. Correct statement. Located in vascular tissue bundles in the stem and leaves. This is correct as well. So, C looks correct altogether. And D, they had elongated cells with lignified cell walls with pits at intervals associated with companion cells of the roots only. No xylem ever has companion cells. They're dead. They're not associated with anything. So C is the only correct option. Number 29. Which properties of a lignin are important for the function of xylem vessels in the stem of a tall plant, such as a tree? It is inflexible, so it does not bend easily. Yes. kind of like cellulose. This one, statement one is kind of outside the syllabus. So you have to really look at the other ones to infer whether it's correct or not. So let's look at the other options. It is not permeable to water. Yes, it forms a watertight pipe. It is strong to resist collapse under pressure. Yes, one of the functions of lignin is this. It is weaker addition to water molecules in cellulose. No, they have approximately the same adhesive quality. So 2 and 3 are correct. So this this is correct. And 1 is also incorrect because 1 is also correct because it's like cellulose. So A is the correct answer. Number 30. Which row is correct for sucrose concentrations and the water potentials in a source and a sink in a potato plant at 12 p.m.? So first, let's revisit one of my notes, which is the transport of sucrose from source to sink. Okay, great. So as you can see over here, sucrose is loaded from the sieve tube, from this phloem into the sieve tube elements. And when it gets into the sieve tube elements, the water potential inside the tube decreases. And as a result, water moves from the sieve tubes. Water enters into the sieve tubes by osmosis down the water potential gradient. And if the water concentration inside the sink tube increases, the hydrostatic pressure also increases because volume increases, and this leads to the creation of hydrostatic pressure gradient, which moves solids and stuff down from the source to the sink. So the water potential at the source is lower, while the water potential at the sink is higher. This helps create a hydrostatic gradient. And well, the sucrose concentration in the source is obviously higher than sucrose concentration in the sink. Otherwise, sucrose won't go there, right? So B is the only correct option here. Number 31. The diagram shows one possible way in which sucrose may be loaded into a sieve tube element. So, which steps require ATP? Number 1 requires ATP because if photons are moved against their concentration gradient using energy of ATP by the process of active transport. So 1 does require ATP. Number two, photons move down their concentration gradient by facilitated diffusion, while sucrose molecules move by against their concentration gradient through co-transporter proteins. So 2 does not require any energy, does not require energy, and thus it does not require ATP. So 2 incorrect. And 3 is just the passive movement by diffusion. So 3 is obviously not correct because diffusion does not require energy and thus not ATP as well. So 1 is the only correct answer. C. C is the correct answer. Number 32. Which statements about arteries are correct? Artery was to resist high blood pressure, correct? Arteries pump blood out of the heart. No. Arteries carry blood. Carry blood. So this is obviously incorrect. Blood in arteries has the same flow rate as in veins. No. Blood in arteries has higher velocity. Higher flow rate as higher pressure. So this is obviously incorrect as well. Then the blood pressure, the pulse in arteries is the result of a surge in blood that causes expansion of the artery walls. So suddenly the high pressure blood goes through the arteries and this causes the artery walls to expand against themselves, and this causes this pulse that we hear. So 4 is correct. Then number 5. There are semilunar valves at the junction of arteries with the heart. Yes. There is pulmonary valve. There is pulmonary valve which is present between the right ventricle and pulmonary artery. And then there is the aortic valve. It is present between the left ventricle and the aorta. Okay, so options 1, 1, 4, 5 are correct, which gives us C as the correct option. Number 33. What happens during ventricular systole in a mammalian heart? The atrioventricular node transmits an electrical signal to the apex of the heart. Not really, because firstly, the atrioventricular node transmits pulses to the Purkinje tissue, and then the Purkinje tissue transmits it to the apex, which is the base of the heart, and then the ventricles contract base upwards. So this is incorrect. The pressure in the ventricles drops below the pressure in the atria. Okay, for this we need to look at our notes. Yes, this is the correct diagram. You will be required to identify which valves open where. So as you can see in my diagram over here, ventricular systole is this part, and in ventricular systole, AV valves are closed while the semilunar valves are open. AV valves closed while the semilunar valves open. So this is correct. The pressure in the ventricles drops below the pressure in the atria. No, this is incorrect, because if this was true, then blood wouldn't even flow from, then blood wouldn't be pumped out of the ventricles. The ventricles will be totally relaxed. But the opposite happens. Ventricles are contracted and we pump blood out of the ventricles. So 1 and 2 are wrong, which gives us 3 as the correct option, and thus D as the correct answer. Okay. Number 34. A shortage of protein in the diet of children can lead to a disease that causes excess tissue fluid to build up in the abdomen. What explains why a tissue fluid buildup of excess tissue fluid can occur in the disease? Number 8. Option A. A shortage of plasma proteins decreases blood water potential. It actually increases because solid positive increases. Less tissue returns to the blood. Once incorrect. A shortage of blood plasma proteins increases blood water potential. This is true. And as a result, if it is increased, then the concentration gradient between blood and tissue fluid decreases. As a result, this can be the correct answer. If the concentration gradient between blood and tissue fluid decreases, then less tissue fluid would return to the blood. Then, option C, a shortage of protein in the diet causes weak cardiac muscle, reducing hydrostatic pressure. This is totally unrelated, so this is incorrect. D, a shortage of protein in the tissue fluid decreases water potential. No, as I said already, it's increasing because the solid concentration decreases. So D is incorrect as well. Number 35. Red blood cells contain a molecule known as 2,3-biphosphoglitated, 2,3-DPG. When PPGD binds to hemoglobin, higher partial pressure of oxygen is needed to bring about 50% saturation of hemoglobin with oxygen. This is kind of like the Bohr effect, which is basically the decrease in affinity for oxygen when carbon dioxide is present. This causes the hemoglobin dissociation curve to shift to the right because a higher oxygen saturation A higher partial pressure of oxygen is required to reach the same percentage saturation of hemoglobin with oxygen. So this is kind of like the Bohr shift. So let's see the statements. Which statements about the effect of 2,3-BPG are correct? 2,3-BPG in red blood cells causes the hemoglobin oxygen dissociation curve to shift to the right, correct? The binding of 2,3-BPG to the hemoglobin reduces the Bohr effect. Well, this is incorrect because this is actually increasing the Bohr effect. It's something like the Bohr effect. Then number 3, the binding of 2,3-BPG to hemoglobin lowers the affinity of the hemoglobin to the oxygen. Yes, because more oxygen is now required to meet the same saturation of hemoglobin with oxygen. And then number 4, when 2,3-BPG is not present, oxyhemoglobin is less likely to bind with oxygen. No, because you don't know how 2,3-BPG works, right? And also, affinity for oxygen might increase but CO2 also persists, right? And CO2 would make oxygen hemoglobin still unload oxygen in places where the carbon dioxide concentration is higher. For example, in active respiring people. So number 4 is incorrect. And thus only B is correct, which is 1 and 3. Number 36. Carbon dioxide is continually produced by respiring cells. It diffuses into red blood cells and dissociates into hydrogen ions and hydrogen carbonate ions. Hydrogen carbonate ions will diffuse out of the cell in exchange for chloride ions in the chloride shift. So let's look at my diagram for this procedure over here. As you can see in my diagram over here, what is happening is that the carbon dioxide combines with water to form hydrogen carbonate, and this is catalyzed by the carbonic acid. Okay, let's start again. Okay, so as you can see over here, the carbon dioxide reacts with water in the red blood cell, and it is catalyzed by the enzyme carbonic anhydrase to form hydrogen carbonate. And then this hydrogen carbonate dissociates to form H plus and HCO3 minus ions, and then the bicarbonate ion leaves the and this bicarbonate ions leave the red blood cells. And if the bicarbonate ions leave the red blood cells, what happens is that the inside of the red blood cell will be positively charged, and we don't want that. And as a result, there is an influx of chloride ions back into the red blood cell to ensure electrical neutrality. So let's look at the options here. It helps to maintain a neutral pH in the red blood cell by acting as a buffer. No, it does not act as a buffer. So what is a buffer? It's something that resists changes in pH. Something that resists changes in pH. So, for example, if something becomes acid, then the buffering action of the molecule causes it to become less acidic by becoming more alkaline. So that equilibrium is reached. So, all in all, A is incorrect. It helps to maintain a balance of positive and negative charge between the cell and the plasma. This is correct. It causes the oxygen dissociation curve to shift to the left, releasing more oxygen. This is incorrect because this has nothing to do with this. It prevents carbon dioxide combining with hemoglobin, allowing hemoglobin to combine with oxygen. Again, incorrect. So B is the only correct answer. Number 37. The photomyograph shows a section through a bronchus. which annotated label is correct? Cartilage, if the airway open, well, the function is correct, but the arrow is incorrect. It's originally supposed to be over here because the cartilage layer is this one. This entire layer is where the cartilage resides. Okay, so C is incorrect, understandable. Elastic tissue stretches when bleeding in. As I talked about this earlier, this layer over here is the cartilage, not elastic tissue. So B is incorrect. D, squamous epithelium allows for efficient gas exchange. No, bronchus doesn't have squamous. It has ciliated epithelium. Squamous is only present in alveoli. Okay, so D is incorrect as well. This leaves us as only A. Yes, the layer just below the ciliated epithelium is where the smooth muscles are present, and smooth muscles change the diameter of the airway. The function is also correct as well, so A is the correct answer. So you also need to know that bronchus has incomplete blocks of cartilage, while trachea has a C-shaped ring. So how do we understand these are incomplete? So look at the cartilage over here. This is one cartilage. This is yet another cartilage. As you can see over here, these are not continuous. And the entire layer has blocks of cartilage, but they are not connected to each other. While in trachea, what you would see is that the entire layer would be a layer of cartilage. Okay, moving on to question number 38. Penicillin is an antibiotic that is used to treat infections caused by bacteria, which events explain how bacteria become resistant to penicillin. The number of bacteria with the allele for resistant to penicillin increases, correct? Resistance to penicillin is a result of a mutation in a bacterium, correct? Then, bacteria with the allele for resistant to penicillin pass the allele to their daughter cells. This is correct as well. Survives because these bacteria have an advantageous trait, and then they survive and reproduce to pass on their advantageous traits to their offspring. On number 4, the mutation for resistance to penicillin is always caused by the presence of penicillin, not CV, not CV, because penicillin causes a selection pressure. So what happens? The random mutations occur all the time, and they cause the penicillin mutations. And survival of the fittest. Basically, some of the random mutations are beneficial, and the organisms which have them are better suited to survive the extreme environment and reproduce and pass on their advantageous traits to their offspring. So survival of the fittest. So this is incorrect. This isn't caused by penicillin. It might happen that the mutation might occur even when there isn't any penicillin around. So number 4 is incorrect. That leaves us as only B with the correct option. Number 39, which statements explain why some vaccine can be taken by mouth, but tuberculosis vaccine has to be injected? Macrophages present antigens in vaccines to stimulate an immune response. Okay, but this does not have any relation with what we're talking about. The TB antigens necessary to produce an immune response are proteins which will be digested in the stomach and the small intestine. This is true because there are protease enzymes present which may neutralize this. And then number 3, there are no B lymphocytes and T lymphocytes in the stomach. This is incorrect. because in fact there are. And if there weren't any T lymphocytes and B lymphocytes in the stomach, why would we even take vaccines orally? There would be no need to do so, right? We would have to take all vaccines by injection, but it's not that. So this is incorrect as well. Just 2 is correct, which leaves us D with the only correct option. Number 14. A student wrote the following statements about the production of monoclonal antibodies. Which statement is not correct? So, in order to tackle this question, let's look at one of the notes I made from the mark form. So, what I did last year is that I made marking notes and book notes from after solving 10 years of question papers. So, these notes are very marking max, and this will help you score marks as well. So anyways, what happens first is that the antigen is secreted into mice to initiate immune response. So a non-self antigen is injected into a mice, and then plasma cells secrete the specific antibody in response to a non-self antigen. This is correct. The selected hybridoma cells divide by mitosis to form a clone. This is correct. And then because hybridoma cells are formed by cancer cells, and cancer cells can divide an infinite number of times, and then T cells are fused with cancer cells to form hybridoma cells. No, this is incorrect. Another name for cancer cells is myeloma cells. Anyways, instead of T lymphocytes is actually plasma cells. Plasma cells which secrete the antibodies necessary. So, since D is not correct, D is the correct answer. And with that, we have finished an entire paper of 9700 AS level biology. If you do like our video, feel free to like, subscribe, and if you have any queries or if you would like to obtain our notes and solve question papers, feel free to check out our Facebook and Instagram pages, as well as our Discord server. Goodbye.